Okay, fantastic. Well, thanks again, Lorrie, for that lovely introduction. And thanks, AJ, for this invitation. And I want to commend AJ for doing really just a phenomenal job of managing the journal. And really, it's really quite beautiful to look at. And the content is really quite extraordinary. So it's really a great achievement. So I'm going to talk, as Lorrie mentioned, about the lysosome. My lab actually for many years was very interested in mitochondrial function. But we had a sort of late life organelle crisis, and we've become more interested. We've become not more interested, but we certainly become interested in the lysosome as well. And I wanted to go through how that interest evolved over time. So one of the sort of relationships that we've been very interested in trying to understand over the last decade or so is why in every organ there is an age-dependent decline in function and a corresponding age-dependent increase in disease. And so, as an example, in the cardiovascular system, here's just one measure of an age-dependent decline in function. This is looking at flow-mediated dilatation as a function of age. And as you can see, there's some scatter, but basically with time and age, the ability of the vasculature to respond and dilate is impaired. And correspondingly, if you look at disease incidence, there's a reciprocal relationship that as you get older, obviously the age-related increase in diseases occur. And this is obviously not this relationship of, again, an age-dependent decline in physiological function coupled to an age-dependent increase in disease incidence in that organ system is not unique to the vasculature or the heart, but plays out in every organ, in every tissue in the human body. And so a very simple, but I think really important question is, what biologically connects this decline in function to this susceptibility to disease? What is the mechanistic coupling that links these phenomena together across all tissues? And I think, there's probably more than one answer to that question, but we've been very interested in this concept that autophagy might be that link, that autophagy might be a reason to explain why tissue physiological function is impaired with aging and why there is more susceptibility to disease with aging. And again, this audience is probably very well familiar with this concept of autophagy, first described in yeast, but obviously very well conserved across all evolution, in which damaged contents, be they damaged proteins or organelles like mitochondria, are recognized to be damaged, and then encased in a membrane, the autophagosome, that is formed de novo. And that autophagosome then fuses with the lysosome, and which allows the contents of the autophagosome to be degraded, and then thereby achieving two purposes. One is the removal of those damaged contents from the cell, thereby creating, maintaining homeostasis. And that degradation also provides biosynthetic intermediaries to allow the synthesis of new molecules, and thereby the cell can be maintained in sort of what I would view as sort of a youthful state. So the reason this is really linked to aging, I think, really comes not as much from mammalian systems, but from more simpler organisms, and particularly work in C. elegans. And so here is just sort of a summary of a number of pathways that have been shown in the worm predominantly, but in other organism systems as well, to increase overall lifespan. And these are very, very diverse stresses or interventions, from dietary restriction to, in the worms, the removal of the germline, to manipulations of mitochondrial function, to changes in IGF signaling or TOR signaling. And in an attempt to understand what could be the common thread, the common mechanisms by which these various disparate life-extending interventions occur, I think the majority of evidence says that perhaps the only single factor that unites all these is that all of them appear to produce an increase in autophagic flux. And again, in simple organisms, by doing epistatic experiments, one could show that, in fact, that not only is this a correlation, but that this increase in autophagic flux in these simple organisms is actually required for the life extended effects. So therefore, one could almost view, at least in very simple organisms, that life extension really requires the maintenance or the augmentation of autophagic flux as we age. Is there a decline in autophagy as we age in more complex organisms like mammals? And so a few years ago, we wanted to develop systems to be able to at least begin to measure this in living organisms. And one of the things that we've done is to develop reporter mice that allow us to do this. And in this case, this mouse, which we call the mitokema mouse, allows us in this case to visualize a subset of autophagy, in this case mitophagy. And I won't go through the details here, just to show you the visual here of what this looks like. So I hope you can see my pointer. And here we're looking in the hippocampus of a young animal, and the orange fluorescence here really is a readout of mitophagic flux. And you can see in the hippocampus, which is really the sort of the anatomical location of where learning and memory occurs, that in the young animals, there's quite robust evidence of mitophagy occurring in these animals. And in that same region, in the old animals, one can see that although the mitochondria still exist, which is the green fluorescence, the orange fluorescence which is an indication of mitochondrial turnover, is dramatically reduced. And so this and other evidence from other reporter mice and other systems suggests that there is really a fundamental decline in autophagic flux as we age. And one of the questions is why is that occurring? And again, I won't go through all the data, but I think a lot of evidence points to the fact that there's an intrinsic defect in lysosomal function as we age. Again, the lysosome is that terminal acceptor of autophagy, the place where that's sort of the last stage of where the autophagosome fuses at the end. And the maintenance of lysosomal function appears critical in order to maintain autophagic flux. And there's a variety of evidence that suggests that as animals and organisms age, the ability of the lysosome to maintain its normal homeostasis is impaired. And so that got us thinking in terms of what we had always been interested in, sort of quality control pathways. And for instance, what controls the quality control of mitochondria. But we really had never thought about what controls the quality control, what are the quality control pathways that maintain lysosomal function. And so we sort of became interested in that question. And so first we asked what's already known about lysosomal quality control. And before we started our work, there had already been some work to understand what happens when the lysosome is damaged, how is it repaired or taken care of. And one process that was described very early on is this process of lysophagy, which is sort of counterintuitive, but in this case, whole lysosomes can be removed by the process of autophagy. So in other words, an autophagosome, instead of delivering contents to the lysosome, actually encompasses a damaged lysosome and degrades it. So that potentially removes the damaged lysosome from the cell. So that is a process that occurs, but it's a relatively rare and sort of terminal event for the lysosome. When the lysosome is damaged, but is thought to be capable of repair, another pathway was demonstrated to occur. And this is a family of proteins known as the escort family of proteins. And they're involved in many processes in the cell involving repair of membranes. But three separate groups in 2018 showed that when lysosomes are damaged, that one process that repairs that damage is that these escort proteins are recruited en masse to the damaged lysosome. And together, they're involved in repairing the damaged lysosomal membrane. So just to understand the sort of experimental paradigm that's used here. So again, the lysosome is this organelle very, very acidic, right? It's got a pH of about 4.5 or 5. So lots of damaging acid in the lysosome. And it's got filled with lots of damaging enzymes, which are sequestered in the lysosome under normal conditions. But in all of those examples where lysophagy or the escort proteins were shown to be recruited, what the scientists did was to damage the lysosomal membrane. And there's a variety of ways to do that. And that lysosomal membrane permeabilization, damage to the lysosomal outer membrane, is sensed by the cell to either remove the lysosome by lysophagy or to repair it by these recruitment of the escort subunits. Because if it doesn't do that, there is a release of all of these damaging enzymes like cathepsins, which can rapidly induce lysosomal dependent death in the cell. And classically, what's done is to use this detergent, what we abbreviate as LLOME, which is basically a small peptide that can go accumulates in the lysosome. It's cleaved by cathepsins, which then allows it to polymerize, and that polymerization pokes holes in the lysosomal membrane and induces this repair process. So again, just going back to those papers that were published in 2018 when we began our work, we just wanted to see, in fact, were those reports, could we reproduce those reports? And so here what we're doing is looking at the cell under baseline conditions in this sort of top row, and then immediately after inducing lysosomal damage, again with this chemical LLOME. And what we're doing is staining by immunohistochemistry for these escort subunits. And here are just two of the many escort subunits. And so you see very little staining, punctate staining under basal condition. But then within minutes of damage to the lysosome, you see this incredible punctate form in the cells, in the cytoplasm. And you can do a number of studies to show that these punctae are actually damaged lysosomes. And each of these dots corresponds to a damaged lysosome in which, in this case, this escort subunit is recruited. And again, this is sort of used by many as a marker for a damaged lysosome, the recruitment, this punctate recruitment to the lysosomal surface. So that was all great, but if you actually looked at the data that was published in 2018, you came up with a conundrum. So here is the work that was published in Science by one of those three groups. And what they did is here damage the lysosome with just another chemical that can, like LOME, that can damage it. And they looked at the kinetics of repair. And so obviously when I won't go through the assay, but here's the lysosome was damaged. And you can see that in the control cells, you can repair that damage over a 10 or 12 minute period of time. And if you disable the escort complexes by knocking down one of the critical subunits, what you can see is that the repair is slower, but and less complete. But it's a relatively modest defect, and it suggests that there may be other pathways that are involved in rapid lysosomal repair that are sort of escort independent. Because in the absence of escort repair, there still is a relatively robust ability of lysosomes to repair themselves. And so the question that we wanted to ask is, what are the other pathways that repair lysosomes that don't involve the escort machinery? And so this is where I had a really incredibly talented person join the lab, Jay Tan, who took this on. And the strategy that we, and Jay now is actually a faculty member here at our institute and has his own lab and is pursuing many of the things that I'll tell you about over the next 15 minutes or so. But the strategy that we used, again, we had no really idea what this pathway was. And so we thought we had to take an unbiased approach to try to describe what it could be. And so the strategy that we used was to engineer a cell line to express this sort of fusion construct where we used the LAMP1 targeting sequence to the lysosome, a GFP moiety just to see it. And then on the outside of the lysosome, facing the cytosol, we express this enzyme called turbo-ID. Now, turbo-ID, for some of you may hopefully are familiar with, others may not be, but it's actually a bacterial enzyme that acts as what's called a proximity biotinylase. So in essence, what it does is that it adds biotin to anything that's near it, in this case, roughly 10 to 15 nanometers away. And so biotin, as you know, is a small molecule that's sort of like niacin, and it can be covalently attached to proteins by this turbo-ID strategy. And the advantage of that covalent attachment of biotin to the protein is that it allows you to then rapidly purify biotinylated proteins using another molecule called streptavidin. And the biotin streptavidin affinities are sort of like antibody-antigen affinities, very high. And so you can rapidly purify proteins that are in one step and identify them by mass spec using this biotin tag. And so what we did here, what Jay did, is to engineer this cell so we could essentially biotinylate the lysosomal surface. And we did this really under two conditions, under normal conditions, under basal conditions, and then 30 minutes after we damaged the lysosomal surface. And then we asked what was different about the proteome on the surface of the lysosome under normal conditions versus under damage. And the answer was quite a lot. And as you can see, we published this a few months ago in the journal Nature. And so here's an alignment of what we observed on the surface of the lysosome, either under basal conditions shown in blue, or proteins that were enriched after lysosomal damage shown in orange. And the good news is that we got a lot of hits here, and shown in blue are all the escort subunits, right? So that was a good sign because we knew that those proteins, as I had showed you, and those other groups had demonstrated, we knew that those proteins were in fact recruited to the surface of the lysosome. But we weren't actually interested in those proteins because that pathway had already been described. But what Jay noticed were these three proteins in green, this enzyme PI4K2 alpha, and these proteins ORP9 and ORP11. Those three proteins were recruited at very high rates to the surface of the lysosome with damage. And those three proteins suggested a pathway because PI4K2 alpha is a lipid kinase, and it generates a lipid called PI4P, which I'll explain in a second. And ORP9 and 11 are proteins that are known to bind PI4P. So the fact that these three proteins were recruited to the lysosome suggested that this pathway, a lipid kinase PI4P dependent pathway, may be important for lysosomal repair. And no one had really described that before, so we thought that might be worth pursuing. So again, if this is true, this predicts that, in fact, we should see PI4P on the surface of damaged lysosomes. And so that's the first thing that Jay did. And again, I won't go through all the details because, again, this gets a little bit involved in sort of lipid kinase signaling. But there's really some very nice tools to look at intracellular phospholipids, phosphoinositides in cells. And basically these employ domains of protein that are known to specifically bind to those phosphoinositides that are hooked up to fluorescent moieties. And so here's one example of a protein that's known to bind PI4P that's used as a reagent to localize the PI4P in the cell. And maybe just because it's small, I blew it up here so you can see it a little bit more clearly here. So here what we're looking at is a blow up in the cytosol of escort subunit shown in red or PI4P shown in green. And under normal basal conditions, you really don't see anything. But then 10 minutes after damage, you see again these punctae, which I showed before for that label with these escort subunits, which represent damaged lysosomes. And you can see using this PI4P probe that each of these damaged lysosomes now has on its surface, now stains positive for this PI4P probe. And there's essentially a 100% overlap of these two markers, suggesting that every damaged lysosome now is decorated with this lipid PI4P that is generated de novo on the surface of the lysosome. And again, I won't go through all the details, but that's sort of shown quantitatively here. So again, under normal conditions, we see no PI4P on the lysosome. But then immediately after damage, you see this huge increase in PI4P on the surface of the lysosome. Now, if you CRISPR out this kinase that we identified that is recruited, that PI4P on the surface of the lysosome is gone, basically. So it definitely requires this PI4K2 alpha in the cell to generate this lysosomal PI4P. If you take these knockout cells and you put back the kinase, you can restore this. But if you put back a kinase dead mutant of PI4K2 alpha, it's unable to do that. So that suggests that you need this kinase and you need its kinase activity to have this PI4P on the surface of the lysosome. So just to summarize what I showed you before is that the lysosome is damaged. You recruit this kinase to the surface of the lysosome, and this lysosome then generates this new novel lipid on the surface of the lysosome that's not here. There is phosphoinositide on the surface of the lysosome, but now there's a new phosphoinositide, PI4P, okay? And so this is really nice because in a way, this identifies, again, you have hundreds of lysosomes on your surface, in your cell, and you only want to repair the damaged ones. And so basically what we're arguing is that this lipid generated on the surface of the damaged lysosome and only on the damaged lysosome provides a marker for the cell to tell that this lysosome is damaged because it now has PI4P on the surface that it never had before. So how does the cell recognize and what does it do now that PI4P is on the surface of the lysosome? First, is that important? First, before I show you how it recognizes it, I want to just prove to you that in fact that you really need that PI4P to really repair your lysosomes. And so again, there are multiple ways at looking lysosomal repair. One of this is an assay called using this reagent called pH Lorrie, which is basically a pH sensitive dye. And so under acidic conditions, it doesn't fluoresce. When the lysosome gets alkalized, you can see fluorescence. And then when it gets repaired, that fluorescence goes away. And similarly, there's another assay we've used in the manuscript that looks at lysosomal pores as evidenced by galectin-3 binding. So I just want to show you that this generation of PI4P is absolutely essential for lysosomal repair. So again, we're now using this pH Lorrie assay, which again measures lysosomal pH. So you damage lysosome, there's this rapid alkalinization of the lysosome, again, because the membrane is damaged. But under normal conditions, you can rapidly repair that and restore the pH of the lysosome over the next few hours. If you CRISPR out these that kinase PI4K2 alpha, you can see that now the lysosomes are very impaired in their ability to repair the damage, and now the half-life extends to from about 1 hour to about 6 to 10 hours. If you put back the kinase into these knockouts, you completely rescue the phenotype. And again, if you put a kinase defective mutant back into these cells, you're unable to rescue. Again, so this suggests again that this generation of PI4P is critical for the repair of the lysosome. So again, how does this do it? Again, this is really complicated, so I want to walk you through it. So again, you damage the lysosome. I've showed you mostly data with this agent, but in the manuscript, we show a variety of different ways, both genetic and chemical ways, to damage the lysosome. All recruit this 2-alpha. We believe that it's because that this is through the release of lysosomal calcium as a signal, but we don't completely understand all the signals that allow 2-alpha to be recruited to the surface of the lysosome. But again, this recruitment then is able to activate the kinase and generate this new lipid on the surface of the lysosome, PI4P. And the question is, what happens next? How does the cell respond to this? And again, we had a key already because, again, as I mentioned, we pulled out these two other proteins, ORP9 and ORP11, as also in our initial proteomic screen. So what do these proteins do? So this actually is, again, quite complicated, but there's a lot of these proteins in your body. And some of them are listed here. And these, again, evolutionarily conserved from yeast to humans. But to simplify these proteins, they have sort of three common elements that are part of all of them. The first is that they have this PI4P binding motif, as shown here in green. The second thing common to all of them is a motif that allows them to bind to the endoplasmic reticulum. And a third domain, shown here in red, is a lipid transfer domain. And this transfer domain really comes in two flavors, either transporting cholesterol or transporting the lipid phosphatidylserine. And so those three domains really define the three functions of these proteins. First, they act as tethers. So they bring together membranes. One is the endoplasmic reticulum. The other is a membrane containing PI4P. And so they do that by these two different domains here in the protein. And once these two membranes or organelles are brought close together, they exchange lipid. They exchange PI4P for either cholesterol or phosphatidylserine. I know that's quite complicated, but let me just show you graphically here what it would look like. So again, the prediction from the domain structure here is that you have this damaged lysosome, which has PI4P on it. And these OSPP proteins on one domain can bind PI4P on the lysosome. On the other domain, they can bind the ER through this ER fat domain. And then once these two organelles, the ER and the lysosomes, are tethered together by these ORP proteins, then these proteins do one more thing. They take this PI4P on the lysosomal surface and exchange it for phosphatidylserine on the ER membrane. And so they bring PI4P to the ER and bring phosphatidylserine to the lysosome. Okay, so if this model is correct, it would predict that with damage, the endoplasmic reticulum, the ER, and the lysosomes should be tethered together. And so Jay asked, does it, oh, so sorry, excuse me. Just to make it a little bit more complicated, although we got ORP9 and 11 in our initial screen, when we went back and looked at our proteomic data, we actually realized that there were 4 of these family members that were recruited, 9, 10, 11, and OSPP. And we could show both by the mass spec data and by Western blot that they were in fact recruited to varying degrees when we damaged lysosomal membranes. Okay, so back to the question is, if this model is correct and these OSPP proteins are recruited, do we see evidence that the endoplasmic reticulum and the lysosomes interact when the lysosomes are damaged? And I really think that these images are quite pretty. And so I wanted to show them sort of by themselves. And so here what we've done, Jay has done, is to look at the cell under basal conditions, again, without this lysosomal damaging agent, or 10 minutes after lysosomal damage. And in green, what we've done is outline the endoplasmic reticulum. In red, what we've done is outline the lysosome. So under basal conditions, you can see that there is some touching of the lysosomes with the ER. There is some interaction, but it's quite modest, basically, and sort of just a handshake at best. But it becomes a full-on hug, I think, once the lysosomes are damaged. And so you can see here that 10 minutes after damage, the green, which is the ER, is wrapping around the lysosome, completely surrounding the lysosome in many cases, and basically uniformly doing that all throughout the cell, basically, because we've uniformly damaged all lysosomes. So that's shown schematically here. Again, sort of under basal conditions, there's sort of a close approximation of the ER and the lysosome. But then with damage, again, there's just this incredible wrapping around of the endoplasmic reticulum to the damaged lysosome. Okay, and so genetically, what we could able to show is that this pathway that I've described of PI4P generation and ORP recruitment is absolutely essential for this wrapping. So again, when we look at does this occur in wild type cells, you can see that over 90% of the lysosomes have this dramatic ER lysosomal contact. If you knock out this kinase that now blocks the generation of PI4P on the surface of the lysosome, you completely abrogate those contacts. If you put back the wild type kinase in the knockout cells, again, you restore it. And again, it requires the kinase activity because if you put a mutant form of PI4K2 alpha in, you cannot rescue it. So you need this kinase to do it. And you need these ORPs. And again, as I mentioned, there are multiple ORPs recruited. And this really was a heroic part on Jay's part, because what he found was that you actually needed to make a quadruple knockout, CRISPR knockout of ORP 9, 10, 11, and OSBP to sort of abrogate this wrapping effect. And you can see here he made 3 different quadruple knockouts, and all of them are defective for this phenotype. But if you just knock out 1, 2, or 3 of them, the cell is okay. But really, it really required that everyone that was recruited needed to be knocked out in order to block this effect. So again, I realize this is complicated, but what I've tried to show you so far is damage, kinase recruitment, PI4P generation. This PI4P then serves as a signal to the cell that the lysosome was damaged and allows the recruitment of these tethering proteins. Now, these tethering proteins then allow this damaged lysosome with PI4P on its surface to be bound on one end and the endoplasmic reticulum to be bound on the other end. And so therefore, it brings this damaged lysosome in very close proximity to the endoplasmic reticulum, in such close proximity that it now can exchange the PI4P on the damage of the lysosomal surface and now deposit phosphatidylserine on the surface of the lysosome. Okay, so we've made some progress here. And so the question is, what's next? Excuse me, because if you've been keeping score here, we haven't really done anything to repair the lysosomal membrane. We haven't really, as the lysosome membrane is damaged, it needs lipids to repair that membrane damage. But we've really not delivered any lipids to the lysosome. We've made new lipids. We've made PI4P. We've exchanged that PI4 for PS, but we haven't. All we've done in a way is change PI to PS. We haven't really added any lipids to the lysosome. So that suggested that this PS somehow has to stimulate bulk lipid transfer in a way. And so the question is, what could do that? And we searched for potential answers. And what we've came upon is a protein called ATG2AB, ATG2, which comes in sort of 2 genes, A and B. And this protein, as you can tell from its nomenclature, is really an essential autophagy gene. And so, as many of you may know, ATG2 is involved in forming the autophagosome. And it does so by binding to, on one end, to the endoplasmic reticulum, and on the other end to the pre-autophagosome. And it allows the delivery of lipids from the ER to the expanding pre-autophagosomal membrane. And this really is essential as a lipid transfer protein to generate the autophagosome in cells. And there was a hint in the literature to suggest that the activity of ATG2 was stimulated by the presence of phosphatidylserine. And so this really was a great candidate for the next step in our pathway because it had the 2 properties we were looking for. One, it was allowed for high volume lipid transfer, and two, its activity was stimulated by phosphatidylserine. And so again, I won't go through all the detail, just to show you that we think that this represents the final step in this pathway. And here's evidence, some of that evidence. So we could show that that ATG2 was recruited very dramatically and quickly to the surface of damaged lysosomes. In an in vitro assay, we could confirm that its activity was stimulated by the presence of phosphatidylserine. And here what Jay has done is use liposomes, a donor liposome and an acceptor lysosome. And so you can think of this sort of as the endoplasmic reticulum and this as the lysosome. And we add recombinant ATG2A here to the mixture and look for lipid transfer between these two liposomes, but as I can imagine, you can imagine sort of two organelles. And you can see that only if there is phosphatidylserine in the acceptor liposome is there any degree of lipid transfer. So again, this suggests that the ability of ATG2A or B to transfer lipids between, we think, the ER and the lysosome is really dependent on phosphatidylserine in the damaged lysosome. And ATG2 is absolutely required for lysosomal repair. And again, I won't go through the details, but again, we've knocked out either both ATG2A and B, and the lysosomes are unable to repair. And when we reconstitute that back, they are. So hopefully I've walked you through a really complicated pathway, but we think an important one that really has three components for repair. And again, step one is the recruitment of this kinase PI4K2 alpha to the surface of the lysosome. This is a calcium-dependent event, and this generates this lipid on the surface of the lysosome, which acts as a danger sign or a damage sign that marks that lysosome as needing repair. It also serves to recruit this family of OSBP proteins, which then bind on one end to that PI4P on the lysosomal surface, on the other end to the ER, and bring the lysosome and the ER in close proximity. So they tether the damaged lysosome to the ER, and then allow for the exchange of PI4P for PS. Now, as PS accumulates on the surface of the damaged lysosome, it allows for the recruitment of ATG2, which is a bulk lipid transfer whose activity is stimulated by the presence of phosphatidylserine, and then allows, again, now for the direct repair of the lysosomal membrane through ATG-dependent PS-stimulated lipid transfer. The good news about coming up with a new pathway is you can name it anything you want. And so in deference to our city and our university, we called this the PIT pathway, which luckily the Nature editor allowed us to get through, and which stands for phosphoinositide initiated membrane tethering. So that's the first and second step, and lipid transport, which is the third step for rapid lysosomal repair. And so we're obviously very interested in trying to understand how this pathway is involved in a variety of age-dependent diseases. And as I mentioned, Jay, who did this work, has his own lab and is pursuing many, many aspects of this. But one of the things we want to do is try to understand the in vivo implications. And we think that there might be some important ones. And so I'll just, I'm sure not many of you have read this paper by Adolf Hanover in 1842, but he was actually a Danish pathologist, made a lot of interesting pathological discoveries, but one of which was describing the pigment lipofuscin, which, as you know, accumulates in older tissues and is sort of a marker of aging. And he made this observation, as I said, in the 1840s. About 10 years ago, a group made a gene trap mouse that knocked out this kinase 2 alpha that we described as the first step in this PIT pathway. And those mice rapidly die from an age-related aging phenotype, predominantly from a neurodegenerative phenotype. And when they analyzed the brains of those animals by EM, they noted this massive accumulation of lipofuscin in the lysosomes of those animals. And so at the time, they didn't know the role of 2-alpha in lysosomal repair, and they really didn't have an explanation for this observation in their description of this mouse. But we believe that this represents potentially an explanation for Professor Hanover's early observation, and suggests that this PIT pathway may be really important for a variety of for regulating age-dependent decline in the lysosome. And we're obviously now making conditional mouse models to see if we can understand that a little bit better. So let me just summarize this work. We believe that lysosomal activity and quality control declines with age. Other pathways have described lysophagy, which was a relatively slow pathway, and escort-dependent pathways, which are a rather fast pathway. We don't believe the data suggests the escort pathway represents the total pathways by which lysosomes are repaired, and therefore we search for another mechanism. And what I hope to describe to you is this new pathway, which we're calling the PIT pathway, which we actually believe is the predominant pathway by which lysosomes are repaired acutely. And obviously, we're really interested in understanding the therapeutic implications, which hopefully will come from more in vitro studies as well as in vivo studies. So I went too slowly. I had a whole other part about therapeutics, but I think rather than rush through that, I will stop here if that's okay, AJ, and turn it over to the audience and the panel for any questions.